Hello, hello, welcome back. This is going to be part 2 of chapter 1. When we last left off, we took a look at what all cells have in common, how they can be very diverse, but yet they have unity. And we also definitely discussed some of the characteristics of a prokaryotic cell. So what we're going to do in part 2 is we're going to explore the world of a eukaryotic cell. Alrighty, so a eukaryotic cell, its main difference is that when we look at its cellular composition, it will be able to have a nucleus. And the nucleus is where it's going to be housing its DNA. And this is very important because in addition to actually storing the DNA, the nucleus will take on almost a type of control center role where it will be able to regulate which pieces of the DNA are utilized. Now, I do want to point out that within the nucleus, the DNA is housed and it doesn't leave. And that is one of the main reasons why the cell has to do transcription, meaning that if it's interested in a particular gene, that it will have to find that sequence of DNA within the nucleus, and then it will do transcription to make a copy of it in the form of RNA. RNA will be able to leave the nucleus back and forth, and that is then where you have your messenger RNA, your ribosomal RNA, your transfer RNA that make their way over to the cytoplasm, the middle portion of the cell, where you can then go ahead and do your second step, which is translation. And that, of course, is going to be the synthesis of different proteins by stringing together different amino acids. Inside of a eukaryotic cell, we're also going to talk about the different organelles that you'll come across. And one of the reasons this is so important is because when we look at a size comparison, a eukaryotic cell on average is about 100 times larger than a prokaryotic cell, and its volume is about 1,000 times larger than a prokaryotic cell. So the inside mechanism of it is about 1,000 times versus a pro, and then the outside, its overall size shape, is about 100 times. Now, why do I bring this up? Well, because we know that the larger the cell typically becomes, the less efficient its internal workings are, because the volume, the inside of the cell, that determines the nutritional demand and the waste products development expands a lot faster than its surface area. And the surface area is basically the plasma membrane or the cell membrane, and that, of course, is what the cells utilize to get stuff in and out of the cell. So how can a eukaryotic cell make itself so much larger and still be effective? Well, it's all going to be due to the fact that it has organelles, smaller pieces within the cell that have specific functions, and by doing or utilizing these organelles, it's able to also divide up its energy demands as well as its nutritional and waste product production. So we're definitely going to talk about these organelles and what they actually contribute, and keep in mind that all of these organelles will be depending on the nucleus for their information as to what they'll be synthesizing, and of course when they'll be on versus off. The mitochondria is probably one of the most commonly known organelles, and the mitochondria, we're going to discuss the fact that that is what we rely on for our chemical energy in the form of ATP molecules. We are definitely also going to be exploring chloroplasts. Now, we don't have chloroplasts. You don't find them in animal cells, but you will find them in plant cells as well as some algae plants. Algae organisms. And that is because that is the organelle that helps with photosynthesis, the process of converting energy from the sunlight. We'll also talk about different types of internal membrane organelles, and these are going to be organelles that have their own little membrane that divides it from the rest of the cell, and these will be things like the rough ER, the smooth ER, the Golgi apparatus, and we'll talk about each of their overall functions. We're also going to take a look at the cytosol. So the cytosol is the liquid, and it's really more of a gel, I should say. It's going to be the liquid or gel that fills up the cytoplasm, which is the middle section of the cell. And within the cytosol, within this liquid gel, your different organelles will be floating around. So we'll talk about the fact how it is mostly made out of water, how it's filled with different molecules, how we can utilize its movement because it's not static, to get the organelles to move from one side to the next, but also to move things like molecules or any type of excretions from the inside of the cell to the outside. And we also have to talk about the fact that inside of the cytoplasm, within the cytosol, you will also have a full cytoskeleton that will be housed there. And these will be different proteins and different fibers that will help with overall cell shape as well as cell movement, both externally and internally. And then last but not least, we're going to take a look at where these eukaryotic cells came from. And that most likely they originated as predators. So we'll talk about what evidence there is behind that particular statement. On your right-hand side, you have a picture of one of the most simplistic eukaryotic cells, and that is the yeast cell. And as you can see, it's unicellular, and it is two of them are in the middle of budding, meaning that they're making daughter cells. Here you go. That are about to pinch off and go off. This is actually a form of asexual reproduction that they're doing, meaning that assuming that there's no mutation, their daughter cells have the same genetic information as the parental cells. And this picture is basically here to remind me to tell you that eukaryotic cells can both be found in unicellular as well as in multicellular organisms. There is a wide variety of organisms that house eukaryotic cells. All right, so let's take a look at some of these pictures and let's get some familiarity with the most common organelles that you'll come across in a eukaryotic cell. And it would make sense to start with the most important one, which is the nucleus. The nucleus is basically divided from the rest of the cell by a little nuclear envelope. And the nuclear envelope has little nuclear pores, which you can't really see in the picture, but they're just small little openings. And as I mentioned to you before, inside of the nucleus, you're going to be housing your DNA. And the DNA sits in there relatively uncoiled, but that is where you're going to house all your DNA. That's where you're going to be housing all your chromosomes, which are just basically storage units for the DNA. Now, your DNA is not able to move in and out. So instead, what we're going to be doing is we're going to be doing our transcription, as I mentioned to you before, and you're going to make copies of your genes in the form of RNA. And RNA will be able to go in and out of the nucleus, utilizing the nuclear pores to do so. I should also mention that usually within the nucleus, there is a smaller little organelle, and this one is called the nucleolus. The nucleolus is very important because it's going to be the site where ribosomes are synthesized. Now remember, you need your ribosomes for protein production, so it's very essential. And another thing about ribosomes is that you find them across all cells, whether you're looking at eukaryotic or prokaryotic. Remember in part 1, we said that there's 6 things that all cells will always have in common. They'll always have DNA, RNA, ribosomes, proteins, they'll always have a cell membrane, and they'll always have a cytoplasm. So these ribosomes are being synthesized by the nucleolus in the case of a eukaryotic cell, which is just a smaller little organelle that's housed within the nucleus. Here is a closer look at your DNA or your chromosomes within the nucleus itself. You can see that at this point what we're doing is we're looking at a cell that's in the middle of doing either mitosis or meiosis. How do we know that? Well, because normally when the cell is in its interphase, in its regular day-to-day activity, the DNA is kind of loosely unwind, and it just kind of looks, it's not as visible as you would expect it to see. However, when the cell gets ready to divide, part of what we see happening in prophase, the first step of mitosis and meiosis, is that the DNA molecules start coiling up and they start becoming more visible. And in fact, what they're doing, and you can see them transition from here to the second picture to the third picture, notice how the strings are becoming darker and more visible, and they're basically condensing down to what we like to call the typical chromosome shape that we're so used to seeing. And humans have 46 chromosomes, or we could say 23 pairs, because we inherit half of them from our maternal lineage and the other half from the paternal lineage. So in these pictures, we're seeing our chromosomes condensing and fully forming, and we're also starting to see that the nuclear envelope will slowly start to disappear. And the cells do this because their goal is to take this DNA and basically divide it into 2 batches, or 4 batches, depending on if you're doing meiosis. And this is all done in a way to make sure that the next generation of cells still have access to the DNA. Here is a look at the mitochondria. All right, so let's talk a little bit about the mitochondria. We like to call it the powerhouse, or the FPL of the cell. And the reason we like to do that is because that is where cellular respiration occurs. Cellular respiration is the process of taking glucose molecules and converting the energy that those molecules have into ATP molecules. And humans do aerobic respiration, and that basically means that we require oxygen along the way in this step. Now, as a side product of aerobic respiration, mitochondria will go ahead and generate carbon dioxide as a waste product. So this is one of the reasons why we inhale oxygen and we exhale carbon dioxide, because these 2 gases are so essential for aerobic respiration, which takes place in the mitochondria. Now, not all organisms will utilize aerobic respiration. You have organisms like, for instance, cows that do anaerobic respiration, and that basically means that they will utilize another version of the gas. In the case of the cows, they like to use carbon dioxide. There's also lots of different bacteria and smaller organisms that do anaerobic respiration. And of course, you also have fermentation that can take place, which usually we see in small organisms such as the yeast cells. Either way, I'm getting a little bit ahead of myself. The mitochondria can look different in various organisms. If you take a look on your left-hand side, you actually see a little yeast cell, and what they've done there is they've used fluorescent markings to tag the mitochondria. And its mitochondria is a little bit different because it's filled with these green fluorescent proteins all over the place. Ours does not do that. On the other hand, if you look over to the right-hand side, you see what our typical mitochondria looks like. It has a double membrane, so an outer membrane and an inner membrane, and then it has these little crystals, these little enfoldings inside of its cytoplasm. And we also know that that the mitochondria is unique because it has its own DNA. This DNA is called mitochondrial DNA, put a little M over here, mitochondrial DNA. And what's so unique about this DNA is that you actually only inherit it from your mother. So your mitochondrial DNA is 100% identical to the DNA that your mother has. So if you ever watch like paternity type shows, like, I don't know if you guys watch Maury, or there's a whole bunch of judge shows where people go on and they're trying to establish paternity, what they'll do is they'll take DNA from the nucleus, because that DNA is a mix of maternal and paternal. On the other hand, if there's a case like there's a missing child and the mother steps forward and says, hey, that's my child, what they'll do is, assuming that the child was genetically born to the mother, they'll take the mitochondrial DNA, because from the maternal lineage, it should be 100% match to the offspring. Now, the fact that the DNA has a double membrane, and the fact that it has its own, the fact that the mitochondria has a double membrane and has its own DNA sets it apart from all the other organelles that we find in the eukaryotic cell. And that is then therefore going to be the basis of where exactly did the mitochondria come from? And based on research, scientists believe that millions of years ago, the mitochondria was actually a free-living organism, meaning it completely functioned on its own. It was its own cell roaming around, and somewhere or another it was engulfed by a eukaryotic cell, and instead of being digested for food, it was utilized within its cytoplasmic content because it was able to generate ATP molecules, or chemical energy, which of course was of great benefit to the cell, because the more energy you have, the more items you can utilize, the larger the cell can become. Now, let's go ahead and switch over to the next slide, and let's take a look at this illustration of this whole engulfing process of the mitochondria by a larger cell. So here we go. So we have our little aerobic bacterium, meaning it utilizes oxygen when it does its cellular replication. And as you can see, we have its tiny one, it's floating around, and based on scientific data, what they are establishing is that somewhere millions of years ago, there was an anaerobic eukaryotic cell, so a cell that did not use oxygen, and it came across this little aerobic bacteria, and it did endocytosis or phagocytosis, bulk eating. It allowed the bacteria to be engulfed into its cytoplasmic content, and instead of consuming it, it actually just said, you will now become part of my inner cellular workings. Now, I always like to think of it kind of like Pac-Man, right? So the larger cell ate the small cell. And I know every time I tell this story in class, I always get those looks like, what? There's no way that can happen. How do we know this? But it turns out that there's lots of scientific proof behind this particular explanation. There's so much proof indeed that it's actually been upgraded to a theory. It is called the endosymbiosis theory. And a theory in science is something that has been proven over and over again. So what kind of evidence do we have? Well, our first piece of evidence is that the mitochondria has its own DNA. All the other organelles do not. So its own DNA meant that it can function by itself independently. The second piece of evidence that we have is the fact that it has two membranes. It has its inner membrane and it has its outer membrane. And what we mean with that is that none of the other organelles have a double membrane. So why would we need it for the mitochondria? Well, how about if the internal membrane belonged to the original aerobic bacterium, right? And then the outer membrane was placed there by the cell that engulfed it to now say, you now belong to me. And that is then why we will have a double membrane. And in fact, if you take a look at your illustration, they're over there highlighting the fact that the membrane came across from the eukaryotic cell so that it can claim it for its own cytoplasmic content. And once the mitochondria started fully functioning within the cytoplasm of this eukaryotic cell, notice how it was able to switch from anaerobic to aerobic. And this is really important to point out because if you're using oxygen during cellular respiration, it turns out that is the most effective way of producing the most ATP molecules. And the more ATP molecules you can produce, the higher levels of energy you're able to generate, which means you can do more, you can increase in size, which of course is very essential to a eukaryotic cell because of the fact that it's so much larger than a prokaryotic cell. So it's due to the fact that the mitochondria was engulfed and incorporated that these cells were able to become more specialized, more larger, and that will go hand in hand with the fact that the organism that they were representing also was able to grow and develop. Now there is another organelle that meets the requirements of the endosymbiosis theory. It will have a double membrane. Internal one belonged to itself. The external one was placed there by the quote-unquote host cell. It has its own DNA. However, this organelle we don't have. It doesn't classify. It's not found in animal cells. But you will find it in plant cells, and you will find it in some algae species. And that organelle is going to be located on our next slide. So let's take a peek. It's going to be the chloroplast. There you go. The chloroplast is the second example of the endosymbiosis theory. And what we can do is let's go ahead and take a look at the illustration that's on the right-hand side, and we see our endosymbiotic theory occurring again. And basically what we see happening is that in plants and algae and their ancestors, endosymbiotic actually occurred twice. Once to allow the mitochondria, and then the second time to allow in these photosynthetic bacteria. And as you can see, once again, they were engulfed, phagocytosis into the cytoplasmic content. And then what we see happening is that the membrane is then placed around by the eukaryotic host cell. And because the chloroplast now became a functioning organelle, what we see happening is that it was able to transition from an aerobic eukaryotic cell over to a photosynthetic eukaryotic cell. And a lot of scientists stipulate that the mix of the mitochondria and the chloroplast is one of the main reasons why we not only have such a wide diversity of different plant species, but also how they can vary in size. Anywhere from, having really small plants to having these gigantic trees that will grow for thousands of years. This all has to do with the fact that plants are autotrophs. They're able to self-sustain themselves, and that is all due to the chloroplast. And the chloroplast is actually a pretty impressive organelle because in addition to having its double membrane and having its own DNA, what we also see is that it has these stacks of little structures. You can see them right over here. These little stacks are called chlorophyll. And chlorophyll is actually where all the work for photosynthesis takes place. And it's where you're able to generate the energy from sunlight. So it is a very impressive organelle. And humans don't have it, I'm sorry to say. That is why we're classified as heterotrophs or consumers, because we cannot synthesize our own food, our own energy. In fact, we have to rely on autotrophs and other heterotrophs to take their energy and convert it to use for our body. All right, so beyond the mitochondria, beyond the chloroplast in the case of plants, there are a boatload of different other organelles that have very specialized functions within the eukaryotic cell. As you can take a look on your PowerPoint, you see that I've listed the most common one. And as you can see, we already discussed the nucleus. We know that it has a nuclear envelope, that it houses the DNA. Chromosomes are just coiled up DNA strands, and that it has little nuclear pores, so the DNA stays inside, but the RNA can go in and out of it. We also see that you have the mitochondria, which we've just discussed, the fact that it has two membranes, it has its own DNA, and it does cellular respiration, which is the conversion of your glucose molecule into your ATP molecules. And I also want to say something real quick about the mitochondria. Even though the mitochondria has its own DNA, it fully communicates with the nucleus and will basically respond to the nuclear demands of the cell. So just a little FYI. It has its own DNA, but it does respond to the nuclear demands, since the nucleus is the control center of the cell. We've also discussed chloroplast, which we know that has little chlorophyll stacked membranes, and that is where photosynthesis takes place. So that will be housed, so that organelle will be housed within our plant cells and some of the algae species that are out there. And then we have another one called peroxisome. Peroxisome is what we like to call a housekeeping organelle. It is able to basically go after different types of toxins. Primarily, it likes to go after blood toxins and any type of debris. And when it finds these toxins, it will basically remove it through the use of hydrogen peroxide, H2O2. And it's the same type of hydrogen peroxide that some of us might have bought, at CVS or Walgreens, and we use as like a little topical cleaner whenever we have like a cut or something. Same exact concept, so it makes this little bubbling action within the cytoplasm, and the peroxisomes get their name because of the production of hydrogen peroxide, but that is then how they neutralize and get rid of the toxins. Now, there are also a batch of organelles that are classified as endomembranes. And the reason for that is because they're going to use these little vesicles, these little containers, to basically move items in and out. Mostly out because they're going to be producing them and then releasing it into the cytoplasm. Now, I'll show you these in an illustrated example in just a little bit, but I want to go over them, their function, and then I'll also kind of give you an example of how they all work together. So the first one is going to be the smooth ER. ER stands for endoplasmic reticulum. The endoplasmic reticulum is very close to the nucleus, and what ends up happening is whenever a strand of RNA makes its way over, it can be utilized to produce, certain entities. The smooth ER is known for the fact that it will go ahead and help with the production of lipids, and it can also be used to detox from certain enzymes as well as to metabolize calcium ions. This is just to name its most common function. The smooth ER gets its name from the fact that its outer surface is nice and smooth. There's nothing that you can find on it. Opposite of that is going to be the rough ER. The rough ER, or the rough endoplasmic reticulum, gets its name because the outside surface is completely studded with ribosomes. And because the ribosomes are on the outside, it'll give you a hint as to what the rough ER does. It produces proteins. So protein synthesis is what the rough ER is known for. It will also help with some intracellular transport, and it's also utilized during translation because of the ribosomes that are so closely involved with its structure. Now, smooth ER gives you your lipids, rough ER gives you your proteins. Think of these as individual ingredients to any type of secretion the cell is trying to make. Like maybe the cell is trying to make a hormone, maybe it's trying to make a certain enzyme. It's going to need ingredients. So it gets its lipids, it gets its proteins, and then what it does is it takes these individual ingredients and it brings it over to the Golgi. The Golgi is often called the Golgi complex, or some books will call it the Golgi apparatus. It goes by both names. It is really kind of like the UPS store, because the reason we call it that is it's going to take those individual ingredients of the smooth ER and the rough ER. It's going to sort them, it's going to process them, it's going to package it, and it's going to basically produce the final product that you're trying to generate. So is the cell trying to generate testosterone? Is it trying to generate milk? Maybe we're looking at a lactating mother, right? Is it trying to produce certain digestive enzymes? Whatever its final secretion is, the Golgi apparatus will sort it, process it, and package it. It will put it in a little vesicle, a little container, and that vesicle then comes out of the Golgi and can either be utilized within the cell, or it will go over to the cell membrane where it will do exocytosis, meaning it will bud open and release its secretion through the extracellular fluid or extracellular matrix of the cell. So the hormone will float away in the bloodstream, the milk will go through over to the mammary gland through the ducts, anything that needs to be released by the cell. You also have lysosomes. Lysosomes are another housekeeping gene. What they tend to do is they tend to lyse any type of old organelle, and they'll do this through intercellular digestion. So if there's like a mitochondria that's no longer functioning, or even if there's some debris that comes into the cell that you need to get rid of, the lysosomes will go ahead and do intracellular digestion by taking up the old organelle, the debris, putting it in a little vesicle, a little container, and then inside of the container they'll generate a very low pH, a very acidic environment, and then they'll use that to pull it apart, also with the help of hydrolases, which are different enzymes that can pull apart this old broken organelle or debris. And that is then how you get rid of something that you no longer need, or something that you definitely don't want within the cytoplasmic content of the cell. And then of course we have to mention our plasma membrane. The plasma membrane, or the cell membrane, as some of you like to refer to them, that's going to be the border that separates the internal and external environment of the cell. So it will obviously also will be what anything has to pass through that's trying to get in or out. So as you can see on your PowerPoint, it says we use it for transport, we use it for cell signaling, and we definitely use it for communication because of the fact that it has receptors both externally and internally that can freely interact with the environment that it's in. Also the fact that these little vesicles, these little containers, have to go in and out of the cell membrane, carrying whatever type of secretion or load that the cell is relying on. So how do all these organelles play together? Like what would be an example? Well, let me just give you a little scenario. Every morning when you wake up, your body basically figures out what type of hormones you require, right? And for males, usually they require high levels of testosterone. Females usually require high levels of estrogen. And that's just something that we need for homeostatic purposes. So one thing that we see happening is on a daily basis, your body figures out how much testosterone do I need, how much estrogen do I need. Your body doesn't like to store these hormones, so it produces it on demand, right? Now, where do we get these instructions? The nucleus. So the nucleus is our first location that we're going to go to, and we're going to see that certain segments of your DNA are going to get activated. These are your genes, specific parts within the chromosomes, within the DNA, and these genes will go ahead and initiate transcription. So they can make a copy of themselves in the form of RNA. And this RNA copy now has the ingredient list and the production mechanism to produce our testosterone or our estrogen. So the RNA will freely float out of the nucleus through the nuclear pore, and it will go over to the smooth ER where it will pick up its lipids. It will go over to the rough ER where it will pick up the proteins that are required to produce the hormone. And then finally, these individual ingredients will be brought over to the Golgi apparatus or the Golgi complex, where they are mixed together, sorted together, packaged together, and at the end of the day, you get this nice little vesicle that comes out of the Golgi that is filled with testosterone or filled with estrogen. And these vesicles will migrate over through the cell membrane where they will do exocytosis, and the hormone is released into the bloodstream and it floats over to whatever target cells it requires. And your body now has the benefit of the results that testosterone and estrogen have on these particular target cells. So we always see that the organelles have to get their instructions from the nucleus, and we kind of take it from there. Obviously, the mitochondria relies on the nucleus as well as the peroxisomes as the lysosomes. So it's really a collaborate action, and this is one of the reasons why the cell is able to kind of divide up the labor and can afford to be larger, because it's very energy efficient in how it goes about its day-to-day activity. Here is an inside look at the cytoplasm. Isn't that cool? And like I said to you, the cytoplasm is the area inside the cell. It's filled with cytosol, which is going to be an aqueous liquid, mostly water. It's a little bit of a gel formation. And what we're seeing here is that of E. coli. And all these different colored patches that you see are just different molecules that are floating around. The cytosol is by no means an empty spot. It will have, besides the organelles, it will have lots of other small and large molecules that float around. Things like RNA, things like your ribosomes. So we can't forget about the ribosomes, right? Because they'll help with protein synthesis. Your ribosomes, which are produced by the nucleolus, can not only be found in the rough ER, where they're studying the outside, but they'll also be found floating around in the cytoplasmic content of the cytosol. You'll also have lots of free-floating amino acids, since those are so essential for protein production. You will also have another very special component of a eukaryotic cell that you'll find in the cytosol, and that is going to be the cytoskeleton. Now, the cytoskeleton is basically a production of different filaments and different proteins that will help with things like cell movement, cell division, intracellular transport, and muscle contraction. And there's basically three main types that go into the cytoskeleton. You have the thinnest components, which are your actin filaments. Actin filaments are best known for the fact that their interaction will cause muscle contraction. You also have the largest ones, which are your microtubules. Your microtubules are best known for their role in cell division. They will start guiding the chromosomes towards the middle of the cell in metaphase. They'll start pulling apart the chromosomes into separate batches when you do your anaphase. So their little spindle, some of you might remember the little spindle that helps move the chromosomes around, those are all your microtubules that basically allow the chromosomes to migrate from one side to the next. You also have your in-between element, which is called an intermediate filament, and that one just contributes to the overall cell shape. It helps with the movement of the organelles, and usually what we see with the cytoskeleton elements is that they'll be anchored either towards the plasma membrane so that they can have a pull and tug type of mechanism. Some of them also like to anchor near the nucleus. But either way, they are fantastic because a lot of them will communicate with different motor proteins. Oh, I don't know why that circle went so large. Different motor proteins, and these motor proteins will slide or walk across the filaments and the microtubules, and they'll basically be able to transport any type of secretion down or up, meaning like outside the cell and inside the cell. So it's a very dynamic type of format, the cytoskeletal system, and we are going to be seeing it quite a bit, and we're also going to be utilizing it to explain how we can move organelles from one side to the next, how you can have these little pseudopods or extension of the cell membrane, and of course how you can have things like muscle contraction and movement of the chromosomes during mitosis and meiosis. So it's a very essential part of the overall cell shape as well as movement within the cell. Here is a very simplistic illustration of a eukaryotic cell, and the reason I like it is because if we start on the left-hand side, you can see that they went ahead and they showed you the nucleus, they showed you how your endoplasmic reticulum, notice how this one's nice and smooth, right? It's just one solid color. So this is your smooth ER, and then here we have our little green dots. Those are supposed to represent our ribosomes. So the sections that have, oh actually I'm so sorry, the green dots are meant to represent transport vesicles. My apologies. So in this drawing, they didn't bother to put the little ribosomes. Let me put them for you real quick. Here you go. So all the sections that have these little studs on it, those are going to be your ribosomes, and that will be your rough ER. Remember, your rough ER does the proteins, the smooth ER does the fats. Now, those products are all going to make their way over to the Golgi apparatus. The Golgi apparatus usually sits a little bit further away from the nucleus, more towards the cell membrane, and part of that is because once it has its little vesicles with the final product, it makes it easier for it to be expelled out of the cell if need be. You can also see that we have mitochondria, and usually there are multiple mitochondria per cell. Probably in humans, when we look at our cardiovascular system, if we look at our cardiac muscle, they can house up to 25 to 30% of their cytoplasmic content is completely dedicated to having multiple copies of mitochondria, which makes sense because your heart has such a high energy demand, right? On average, it beats every 0.8 seconds. So it's not unusual to see multiple mitochondria floating around. We also have a mention of the peroxisomes, our little housekeeping genes that will be producing our hydrogen peroxide. They like to aim for blood toxins. And then we have our little lysosomes right over here in yellow, and they will go ahead and take care of any type of damaged or unused organelle, as well as any type of debris that comes into the cell. And they do that by generating a low pH within their little vesicles, and of course different hydraulic enzymes they can be utilizing. Now the picture that is on the right hand side, you can see that the colors have changed, and that's because they want to go ahead and highlight the jelly-like substance that makes up the cytoplasm. So you can see your cytosol here in blue, and you can see how your organelles are kind of like floating around within it. And then obviously we have to label the plasma membrane, since that's going to be our border that will divide the internal and the external working of the cells itself. Now I wanted to add this image in here because I have been mentioning endocytosis or phagocytosis quite a bit, as well as exocytosis. So for those of us who need a little reminder, these are both methods that the cell will utilize to get products into and out of the cell. And what makes this method of transportation so unique is that you'll always use a vesicle. You'll always use a little bit of a container. As you can see, whenever you hear endocytosis, that means to enter the cell. So you can see you have little substances that are on the outside. They'll interact with the plasma membrane, and then the plasma membrane will actually bud off into a little section, creating our little vesicle, and that vesicle then makes its way into the cytoplasm. If you have products that need to leave the cell, primarily produced by the Golgi apparatus, we see that the final products are put in a little vesicle, and that vesicle will then make its way over to this plasma membrane, where it will get incorporated into it, and at the same time it will allow its content to spill outside of the cell. And this movement outside the cell is called exocytosis, like finding the exit. So as you can see, both of them utilize the plasma membrane for their vesicles. The endocytosis will actually shorten the plasma membrane because it's making its little vesicles. But then don't worry, because the cell doesn't get smaller, because at the same rate, usually the exocytosis will contribute its vesicle back to the plasma membrane, allowing it to expand. I also wanted to remind you, for those of us who've done exocytosis before, which I do hope is all of us, endocytosis can be divided into three entities. We have phagocytosis, which means bulk eating. This is anytime there's like a larger molecule or a solid product trying to enter into the cell. So for instance, if we see a little macrophage, a little white blood cell, if it comes across a bacteria cell, it will phagocytose it. It will bulk eat it. If we see that there are liquids or oils that are trying to come into the cell, we call it pinocytosis. A student once told me they think of Pinot Grigio, like the wine, and then every time they hear pinocytosis, they think wine. Wine is a liquid, so you're moving liquid into the cell. I don't know why that analogy stuck with me, but hey, I'm sharing it. And then the last one is called receptor-mediated endocytosis. And receptor-mediated basically says, in order to enter the cell, you actually have to interact with some of the receptors on the cell membrane, and it's only when you fit onto the proper receptor that the process of endocytosis will occur, and you'll bud off a piece of the cell membrane to make your little vesicle to enter. There are certain disease models that actually use receptor-mediated endocytosis. Probably the best example of that is HIV. HIV will interact with your T helper cells, which are a specialized white blood cell, and they will actually match with two receptors that are on the surface of the white blood cell. And when they match to the receptors, it basically tricks the white blood cell to engulf the virus, and that is then how the virus gets access to your genome. The reason I'm telling you this story is a lot of work is being done to develop a vaccine or a treatment that will actually hide the receptors from the virus. And the reason that's so important is because if you hide the receptors, even if you become exposed to the virus, if it can't find the receptors, it can't be endocytosed. And because it's a virus, it needs a host cell to survive. So if it can't enter the host cell, then the HIV virus doesn't do your body any harm because it's then quickly destroyed. So just a little FYI that I like to share. So endocytosis is to enter, and you have your phagocytosis, your bulk eating, your pinocytosis, bulk drinking, and then you have your receptor-mediated endocytosis, meaning that you rely on receptors on the membrane before you can actually enter the cell. And exocytosis does not have a subdivision because everything exits the cell, whether it's a liquid, a solid, etc., and etc. Here is a look at the cytoskeleton network. Figure A, I'm going to switch over my colors real quick, sorry. Figure A is showing you your little actin filaments. As I mentioned to you before, these are very important in things like muscle contraction. Figure B is showing you the largest one. These are the microtubules that will play a role in things like cell reproduction. In fact, you can see they're closely interacting right here with the nucleus, and they'll be moving around the chromosomes any minute now when the cell starts doing either mitosis or meiosis. And last but not least, right here in C, these are little intermediate filaments. And a fun fact is that in cells that have a cell wall, usually you don't find intermediate filaments. They're not needed in that case. Here is another look at our little microtubules, and in this case what it's doing is they're showing you how the microtubules here in green are forming the spindle, and the spindle is what's actually the specific form that's being utilized during your cell division, in this case during metaphase, when the chromosomes will align down the center of the cell. What we see happening is that the spindles will interact with the little centromere, the little midsection of the chromosomes, and it will move them from side to side to get them in the center of the cell. And then once that's done, they will actually start tugging at the chromosomes. And when they tug at the chromosomes, the DNA batch is divided in half. So like in humans, we would have 92 chromosomes that are in the middle. And when they start tugging at the chromosomes, we would see half of them, 46, move upwards and 46 move downwards. And that is then how we're going to divide up our replicated DNA. Once the batches are formed, then you are in your end stage of anaphase. So all that needs to be done is to do telophase, which means that you reform the nucleus, and then you do your little cytokinesis, the splitting of the cytoplasm. And your end result is that you end up with 2 daughter cells if you're doing mitosis, or you end up with 4 daughter cells if you're doing meiosis. But either way, you could see the green as a representation of the microtubules, and you could see one of the essential roles that it plays within cell reproduction, the movement of the chromosomes. Here is an illustration of a typical animal cell. This one is a little bit of what we like to call fibroblastic, meaning that it kind of looks like an extended type of stretched out shape. This is usually what you come across in like connective tissue. And if you look at the internal markings of it, you can see the representation of the actin filaments. You can see your little microtubules are being drawn. You can see the fact that they went ahead and they labeled the mitochondria, the lysosomes, the Golgi apparatus. Look how much mitochondria they drew. That's fantastic. They also went ahead, and in this case, when you look at your ER, your endoplasmic reticulum, notice how some of them are smooth and some of them are studded. Yay! So that's your rough ER. There you go. And then they even went ahead and they drew some ribosomes that are free-floating, which is a proper representation of the ribosomes. They also went ahead and labeled the nucleolus. Here you go. So that is the smaller organelle that you find inside of the nucleus. That is the location where the ribosomes are produced. And then some of them will migrate over to the rough ER, and that's where they'll help with protein synthesis. And some of them will be free-floating in the cytosol. Let's see what else we have going on. We have our Golgi apparatus, I mentioned that before, and then we have our peroxisomes. Fantastic. And obviously we have our cell membrane. Now let's go ahead and take a look at a plant cell. All right, now the first thing you should have noticed is that the plant cell has more of a rigid structure, right? It's not as stretched out or abstract as the animal cell. It's very rigid in its structure. And part of that will have to do with the fact that if you take a look at the plasma membrane, its border, you're going to actually notice that it has 2 things that make up the border. We have our plasma membrane, which all cells have, and then on top of that we have a cell wall. Cells have a cell wall made out of cellulose, which is a carbohydrate, and this cell wall will not only give a lot of protection, but will also be responsible for the rigid shape of the plant cell. If you look over here on your right-hand side, you're going to notice that I wrote down that there are 3 unique structures in the plant cell that you will not come across in the animal cell. So the cell wall is the first one. The second thing is what makes plants autotrophs. They're able to photosynthesize, and that will be the chloroplast organelle. So you can see them pictured right here, nicely green. You can see the thylakoid stacks on the inside. And then the third structure, what do you guys think? Go ahead and take a look at the picture. There's something else that plants have that animals do not have. Da, 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 da. And these are these big old open spaces that you're seeing right here, right? Yeah, so these are called the vacuoles. And what the vacuoles are used for is usually water storage. So as the plant cell absorbs water, the vacuole will fill, and as it fills, it basically presses against the cell wall, and it creates this thing called turgor pressure. That's one of the reasons why if you have house plants, if you water them consistently, they tend to stand up nice and straight, and that's because they have lots of turgor pressure because the vacuole is filled with water. So it kind of presses against and it helps the plant stay nice and straight. On the other hand, if you dehydrate your plants, so like for instance, maybe you give it salt water instead of regular water, which would be horrible, please don't do that. Or if you just forget to water them, maybe you went on a little vacation and you forgot to water your plants. So when you come home, they look all wilted, they look all hunched over. That's primarily due to the fact that the vacuole will completely shrivel up because there's no water inside of it, which means that your turgor pressure goes down and the plant starts drooping over. So the central vacuole is the third item that you will find in plant cells, but you will not find in animal cells. So the chloroplast, the cell wall, and the central vacuole. Now, I do also want to point out that eukaryotic cells can also be found in fungi. So like for instance, your mushrooms, fungi will have a cell wall made out of chitin, which is another, or chitin as some people like to say. This is another example of a carbohydrate. We also tend to have some cell walls within your bacteria, as we were mentioning to you before. So there are lots of examples of cells that have a cell wall, but obviously not all of them have them, including the animal cell. But keep in mind that even though you might not always find a cell wall, you will always find a plasma membrane. Another thing that I want to mention to you about eukaryotic cell is that based on origin, it's been stipulated that most likely the earliest eukaryotic cells were actually predators, meaning that they were out there just kind of scavenging, looking to do nothing but consume smaller cells. And part of that reasoning behind that is the fact that when you look at a eukaryotic cell, its plasma membrane is very flexible, it's very fluid. So like we were talking about the fact how it can make those like pseudopods and extend into its environment, and it's not a rigid shape, so the cell can kind of easily maneuver its overall structure. And obviously when it does so, it can utilize the cytoskeleton to move the organelles and make room within its cytoplasmic content, and also a larger size. The eukaryotic cell is larger than the prokaryotic cell, which all things, if you think about it, the flexible membrane, the cytoskeleton, the larger cell, these are all things that are going to be very important if you are out there trying to consume smaller cells. So those are kind of like one of the reasons why the predatorial history comes into play. And of course, we have our endosymbiotic theory, right? We have proof that the mitochondria and the chloroplasts somewhere along the way were consumed and incorporated within this eukaryotic cell. This, of course, all happened billions of years ago. In fact, if you take a look at your little phylogenetic tree that they're showing you right here, you see that everything started with a prokaryotic cell. That is our simplistic structure of our cell type. It has DNA, but it doesn't have a nucleus. And then somewhere along the way, probably like 3 billion years ago, you had your division between the bacteria and the archaea. And then if we then go up like maybe like 1.5 billion years ago, we see that the archaea went ahead and once again subdivided into the eukaryotic lineage. And from the eukaryotic lineages, we then get a wide variety of different animals, including our plants, our animals, our fungi, and not represented on this chart are our protists, our protista. So things like our amoeba and our paramecium. They also have a nucleus, so they're also classified as eukaryotic cells. The last part of Chapter 1 takes a look at what we call model organisms. These are organisms that scientists utilize in their research to obtain a lot of the knowledge that we were talking about in the chapter about prokaryotic cells and eukaryotic cells. Now, a lot of these organisms are not only cheap to raise, but they'll also have a very quick reproduction cycle, and they'll be easy to maintain. That way they can often be utilized in the lab for prolonged periods of times without actually being too costly on the budget, which is obviously very important anytime you dedicate your life to research. So let's go down this list, and these are by no means all of the model organisms that are utilized, but these are the most common. So starting off with our prokaryotic sample, E. coli is one of the most commonly used bacteria species within the lab, not only for the fact that it grows and reproduces very rapidly and it's easy to maintain within a broad or an agar plate scenario, but also because we know that E. coli will interact with lots of different organisms, including humans, which we can find in our gut specimen, right? So we know it's a bacteria, we know it's a prokaryotic cell, and at the core of it is fantastic because it allows researchers to basically see how this simplistic cell without a nucleus has different fundamental mechanisms of life. Another one that we can look at is baker's yeast. So good old-fashioned yeast. This one is a eukaryotic unicellular organism, and it can basically show scientists the fundamental mechanisms in a eukaryotic organisms. And when you see fundamental mechanisms, they mean things like metabolism within the cell, they mean things like reproduction, they mean things like the cell cycle. So basically, how does the cell maintain itself? How does it reproduce? So just its basic day-to-day activities. There's also a plant model organism. It is a small weed called a ribosomes. I'm sorry, I probably butchered that name. But this is the plant model that we utilize for research. I've actually never worked with this species before. Most of my research was concentrated on using cell lines. So I have some experience with E. coli and with the yeast, but not with this particular weed. But it turned out that this is the most commonly used plant model in research. In animal models, we do have a wide variety of organisms. Probably the most commonly used is Citrocephaly, which are the flies, and they are extensively used for genetics. And that's because their chromosomes are easy to visualize. These fruit flies are extremely easy to maintain. They reproduce very quickly, and you can get a lot of data out of one generation. But obviously, if you're able to mix them and you're able to breed them, they are fantastic for doing things like your Punnett squares and developmental biology. The nematode is next. It's called C. elegans. This worm is so fascinating. We utilize it often for genetics. And one thing that's unique about this organism is that the way that it develops and specializes its cell is very uniform. All C. elegans will have 959 cells when they reach adulthood. That does not account for the gametes, so the oocytes and the sperm that they produce. We're just talking about their regular day-to-day cells. And it's very unique to have an organism that will have such a set number of cells from one generation to the next. So it is a fantastic model to study to see how these cells go from stem cells over to specialized cells. They are also often studied for apoptosis. Apoptosis is cell suicide. It is a very organized method of getting rid of cells that are damaged or are no longer needed. And what's so unique about apoptosis is when the cell is induced to do it, it will actually have no side effects. so it will not harm any nearby cells. It will not cause inflammation in the area of it. So it's a fantastic way to get rid of cells without having any type of symptoms or side effects from it. This zebrafish is the vertebra model that we like to use within animals to study. Zebrafish are used for things like GMO studies. They're very interested to see how the cell behaves during development. And one of the main reasons that zebrafish are utilized, in addition to them being very cheap to maintain, is that their body is actually see-through, transparent, or translucent, as they like to say. It's transparent for the first 2 weeks of development, so you can really see what's happening internally of the young zebrafish. As you go a little bit higher up, these are going to be a little bit more costly. You can also do animal models that are a little bit more complex. Mice tend to be a very popular choice, as well as rats. They are often utilized for genetics, for development. Immunology is a big field that will use rats and mice. So you want to see if there's like a new drug on the market. You obviously don't want to start your experiment on actual living humans. So most of the time you'll start with cell lines, and then you'll migrate over to the mice and rat models before you actually get to the last phase of drug development, which would actually involve humans in your trial. And then last and not least, yes, we actually also look at humans and study them. And you can do humans in many different versions. You can do cell lines, meaning that we have a collection of cells that are originally derived from a human organism, a human sample, I should say, and that are easy to replicate within the lab. So for instance, for my PhD project, I utilized LNCAP cells and MCF7 cells. LNCAP cells are a prostate cancer cell line that was derived many years ago from a patient. MCF7 cells are breast cancer cell lines that was derived from a patient also many years ago that we've been able to maintain in lab, and they're easy to reproduce. So you would start your study on cell lines. You would grow your cells, you would expose them to the drug or whatever treatment you're looking at, and you would see if the cells would, for instance, induce apoptosis, or would their growth increase, or would it have no effect at all. And once you get your data and you're satisfied, you would then move on over most likely to an animal model like the mice and the rats, as I mentioned to you before. And then last but not least, you would move on to actual living human beings, and you would do your human study. So there are varying degrees of how we go about getting our knowledge. Now, a lot of this is possible of doing different model organisms because we see at the core that all cells are composed or have DNA, and all DNA has the adenine, the thymine, the guanine, and the cytosine. And we see even within different organisms that there tends to be a lot of commonality within the genes that they carry. So here, for instance, is an example where you have a picture of a baby and a mouse, and notice how both of them have that little white marking by their forehead, and it turns out that this is actually caused by a defect in a gene called KIT. And this gene is supposed to be utilized for like development, migration, and maintenance of skin pigment cells. And if there's a mutation in another abnormality, it can cause this little white patch to develop. So we can see that both the mouse as well as the human have this gene, and it reacts in a very similar way when mutated. So if you want to come up with a treatment for it, you would start off obviously with your cell lines, but as you move on to your animal models, you can study it within the mouse before you actually go and you try to find your human volunteers, which often are a lot more expensive. And obviously we want to make sure that no human life is harmed when we're doing our experimental trial. I also want to point out that even when you look at different organisms, the actual size of their genome, so how many genes, how much DNA they have, will be varying enormously. And just because an organism has more DNA than another doesn't mean that it has access to more proteins or that it's able to do transcription and translation in a more effective way. And this all has to do in the fact that when we look at our DNA, not all of it is utilized for transcription and translation. So this basically brings us to the conversation of the fact that within our genome, within the DNA, we have what we call coding regions. Coding regions are your genes, so the piece of the DNA that can become RNA, that can become protein. And then you also have vast majority of it, that's what we call non-coding regions, or even repetitive sequences. And these sections of your DNA are just there. They don't do anything. Well, I should say that a lot of people, a lot of scientists are showing that those sections, the non-coding and the repetitive, are really essential for stabilizing and supporting the coding regions. So they do have a role, but they're not contributing to the amount of proteins that we're able to generate. So a large genome doesn't necessarily mean that you increase the amount of proteins that you can produce. It all depends on how much of a coding region you have within the DNA that you're housing. So even when we come over here to our last chart, we can go ahead and we can see that within the different organisms, we have the human, the mouse, the fruit fly, we have our little plant sample, there's the roundworm, the yeast, and the bacteria. Notice how their genome size vastly differs, right? But what we're really interested in are going to be how many protein coding genes do they have access to. And you can see that just because you have a large genome size doesn't necessarily mean that you have the largest amount of protein coding gene, because a lot of that genome can just be junk DNA, non-coding regions, or repetitive. In fact, when they did the human genome study, they basically discovered that for Homo sapiens, for humans, about 98% of our DNA is non-coding. We basically only use 2% of our DNA to produce our protein that we require for our day-to-day functions. We're also able to see when we compare these different model organisms that we have a lot of what we call homologous genes that we share. Homologous genes are genes that have similar sequences, oftentimes very similar if not the same, and they'll also have a very similar function, which indicates that most likely they came from a common ancestor, which would obviously go all the way back to our phylogenetic tree that we saw previously that said that all cells come from that initial prokaryotic cell that started off this journey. All right, everybody, this is where our second part is going to end. Please feel free to email me with any questions, stop by the virtual office hours, and of course, please utilize the discussion sessions if you have any questions, comments, or concern. We're done with chapter 1, so next we're going to be tackling chapter 2, which is going to involve some chemistry. Don't worry, I'll try to make it as pain-free as possible. All right, enjoy the rest of your day.